Good day, and I hope you are having a wonderful day. I would like to welcome you to our General Biology 1 class. Before we continue our wonderful journey into the cell, I would like to introduce our learning objectives. At the end of the session, you will be able to
determine the phases of the cell cycle and their control points, describe the stages of mitosis, and identify stages of mitosis in actual microscopic view samples.
At syempre, bago tayo magpatuloy, I recommend you have a pen and paper handy o kahit anong panulat at masusulatan para makapture o ma-jot down ninyo ang mga impormasyon na importante at para sa activity natin bago matapos ang discussion.
Sa nakaraang lesson, napag-usapan natin ang tungkol sa cell at ang iba't ibang uri nito. Sa ngayon, let's try to recall some facts. I want you to get your pen and paper and answer these quick questions.
Maaari ninyong i-pause ang video na ito ng mga 3 hanggang 4 na minuto para sa pagsasagot. I hope you are done and you did your activity according to the time allotment. Now, let us see if you remembered and retained enough important information from our previous lessons. So, here we go.
So here are the answers to our questions. The first question is, what is a cell? Then we have 3 suggested answers here. The cell is the basic unit of life. We know that. And the 2 other theories says cells come from pre-existing cells and all living organisms are made up of cells. Now there are 2 types of cells. That's our number 2 question. The eukaryotic cell and the prokaryotic cells. They are different from one another because eukaryotic cells have membrane-bound nucleus, such as the animals, protists, fungi, and plants, while the prokaryotic cells do not have membrane-bound nucleus.
Ngayong araw na ito, isa na namang paglalakbay ang ating gagawin sa mundo ng maliliit na tinatawag nating cells. Ayon sa mga eksperto, ang katawan ng isang adult na tao ay may average na approximately 100 trillion cells. Hindi ba nakakalulang numero yan? Well, our cell is us. Ang cells natin ay tayo din. Why do you think this is so? I want you to first answer these intriguing questions before we proceed. 1 representative from the side of this group and 1 representative from the right side of this group, or from this group, will be sharing his or her idea or answer to the question. So, prepare your answer in 1 minute. The teacher in charge will choose the 2 speakers from the 2 groups we have and to share their thoughts. You may now pause this video again when the 2 speakers are sharing their ideas. Well done, guys. Our cell is us because cell in us makes up our entire body, including plants, the other animals, protists, fungi, and bacteria. Many of them carry out genetic materials containing our physical characteristics as well. Pero hindi ba kayo nagtataka? Namamatay din ba sila? Bakit hindi sila nauubos? Paano sila dumadami? Now, these are serious questions that we need to know, just like how much we need to know ourselves. Hindi po ba? The answers to these questions we have now will be answered in our lesson today. Now, the key to the question, how do cells divide and reproduce, can be best explained by studying the 2 types of cell division. We are talking about mitosis and meiosis. In this video presentation, we look closely into the process called mitosis. Maaring narinig nyo na ang salitang mitosis and kadalasan sa mga biology students, dati ay siguradong nakarinig na ng salitang ito. Pero marami pa din sa kanila ang hindi pa familiar sa phases or stages ng mitosis. Aalamin natin yan ngayon. So, mitosis is defined as a cell division process, particularly the division of the nuclei producing two exactly the same daughter cells. Identical po ang napoproduce ng mitosis. So, isa ito sa paraan para mapadami ang cell. Kailangan na dumami ang cell natin habang tayo ay lumalaki at kung tayo ay nasusugatan, for example, at kung kailangan nang palitan ang mga namatay na cell sa katawan natin. Ang mitosis ay parte o bahagi lamang ng tinatawag nating cell cycle. So what is cell cycle? A cell cycle basically tells us how cell duplicates itself. So, most of the time, ang buhay ng cell ay nasa phase na tinatawag na interphase. Makikita ninyo ang interphase ay ang orange na layer sa illustration na ito.
In this illustration, the interface is the longest phase, the orange layer you can see.
Ano ang mga nangyayari sa interphase? Sa unang bahagi ng interphase, ang cell ay unang dumadaan sa G1 phase. Nakikita ninyo, ito yung blue layer.
The G1 phase, which is the blue layer, covers almost 50% or half of the life of the cell.
In the G1 phase, the cell spends time to grow larger. Ang sunod na layer na makikita ninyo ay ang S phase or ang synthesis phase.
You can see that the S phase or the red layer is quite short and it happens quickly.
Nangyayari dito ang duplication ng DNA. Ibig sabihin, kukopyahin ng ating cell ang mga DNA sa phase or stage na ito ng buhay ng cell. Pagkatapos ng S phase ay ang G2 phase.
The G2 phase, represented by the green layer, is very short and it proceeds after the synthesis stage.
Sa phase na ito, ang cell ay patuloy na nag-grow or lumalaki. Now, may mga cells na hindi dumadaan sa prosesong ito. Katulad na lamang ng cell natin sa muscle, yung ibang cell or muscle cells, at ang nerve cells, halimbawa, na hindi nagdo-duplicate. Ibig sabihin, hindi sila nagpo-produce ng kopya nila. Hindi sila nagde-divide. Ang mga cells na hindi na kailangan ng G1, S phase at G2 phase ay dediretso na sa phase na tinatawag na G0 phase, kung saan ay hindi na sila magde-divide. So, ang mga cells ay kailangang mag-divide katulad na lang ng cells in your skin. Matapos silang dumaan sa G2 phase, kung saan ay lalaki sila hanggang sa mature size nila, ay dadaan naman sila sa proseso ng M phase.
From G0 or from G2 phase, the cell now enters mitosis or the M phase.
O ang tinatawag na mitosis. Kung saan naman ang nuclei ng cell ay hahatiin para makabuo ng dalawang magkapareho or identical na daughter cells. Ang resulta ng cycle na ito ay pagkakabuo ng dalawa or two identical o magkaparehong cells. These two cells will again go through the cell cycle and reproduce again like their mother cell. So, if we look at the phases of the cell cycle,
some cells starts at G0, some starts at G1, S, G2, and then mitosis.
para itong orasan kung saan ang mga cells ay gumugugol ng oras where they are spending time in the various stages of their lives. So my question is, in which phase is the cell spending most of its lifetime? Well, you are correct. It is in the interphase where the cell spends most of its time growing, duplicating its DNA and growing some more. Good job! Sa mga laboratory activities natin sa biology, kadalasan tinitingnan o inoobserbahan natin paano magdi-divide ang mga cells sa prosesong tinatawag na cell mitosis or mitosis. Katulad halimbawa ng kadalasang experiment natin sa biology na gamit ang onion cells or ang sibuyas. Kung maaalala ninyo, ang ginagawa natin ay nilalagyan natin ng dye o tina ang sample natin bago tingnan sa microscope para mas makita or mas lalong makita ang mga organelles pati na ang mga chromosomes na nandoon sa loob ng onion cell. In this example, we can see mitosis happening in sea urchin samples. The whole mitotic process is not a one-time and single process. If you look at the video, the yellow structures you can see there are the chromosomes, and the blue structures are the microtubules that help in pulling apart the groups of chromosomes.
So as you can see, the pink or the white structures are the chromosomes, and we have the blue structures as the microtubules separating.
Ngayon, sabi nila, mas madaling maiintindihan ng isang palabas kung panonoorin natin ang pagkalahatan at saka pa isa-isahin at itagpi-tagpi ang mga parte ng pangyayari sa palabas. So, just like the movie, we have seen the bigger picture of mitosis. Tingnan naman natin ang mga nangyari sa movie na ito. So, isa-isahin natin ang mga stages at kung ano-ano ang mga pagbabagong naganap sa bawat steps ng mitosis. Simulan natin sa cell membrane. I will be showing series of pictures according to the 4 phases of cell division. And let's see kung ano-ano ang mga pagbabagong nagaganap sa cell membrane throughout the phases. Tingnan lamang ninyo ang cell membrane at huwag munang pansinin ang ibang organelles sa videong ito.
So as you can observe, nothing is happening in the cell membrane. Nothing, and then it suddenly shrinks and divides.
So, sa videong ito, nothing really happened to the cell membrane hanggang sa biglang nag-pinch off or nag-shrink yung gitna. At ang huli, naging dalawa na ang ating cell. Tignan naman natin ang mga pagbabago ng ibang cell structure. Tignan natin ngayon ang nuclear envelope.
So in the nuclear envelope, nothing is really happening at this stage. You can see, okay, nothing, and suddenly burst into pieces and then gone, and then we have 2 new nuclear envelopes.
Okay, so wala ding masyadong pagbabago at nangyayari. Medyo lumiit ng kaunti at bigla itong nawasak into small pieces hanggang sa ang mga ito ay nawala na. We can no longer see the fragments of our nuclear envelope in this phase. At nang ma-form na uli ang mga bagong cells, the nuclear envelopes reforms, and we also have new nuclei on each side, as you can observe. Alam natin na ang nuclear envelope ay ang membrane na bumabalot at nagpoprotekta sa ating nuclei. Ngayon, may kaunting kaibahan ang cell ng mga tanim or plant cells kasi wala silang centrioles. Ang mga plant cells ay meron naman silang centrosomes. Pero dapat nating malaman na ang nuclear envelope ng ating mga cells ay importanteng structure para sa pagkakaroon ng mga centrioles sa cell. Let us now look at the changes that happen in our centrosome. So, susunod na titingnan natin ay ang centrosome. So, ano ang centrosome? Centrosome is basically made of 2 things. One is the microtubules and 2, the centrioles. Microtubules are the structures which will form into spindles, parang mahahabang sticks, na tumutulong para ma-divide ng cell equally ang mga chromosomes niya sa magkabilang side ng cell. Halimbawa, sa left at sa right. Sa gitna naman ay ang centrioles. Ang mga centrioles naman ang mga nag-o-organize ng mga microtubules at spindles.
So as you can see, the structure of the centrosome, it looks like a flashlight. It's single at first, and then it doubles, naging dalawa, and then it produces spindles and separated the chromosomes.
Sa simula, meron tayong isang centrosome sa cell. Nakikita ninyo sa picture at bigla itong nag-duplicate, naging dalawa. Sa pangatlong pictures, nagfo-form na sila ngayon ng mga microtubules between them. Itutulak sila ng mga microtubules palayo sa isa't isa. And each new cell will have centrosome for each at the final stage. And the process repeats to each individual cells. Ngayon, tingnan naman natin ang pinaka-importante sa lahat ng structures. Tingnan natin ang mga changes o pagbabago sa ating chromosomes throughout the phases. So chromosomes has the genetic information. Sa loob kasi ng mga chromosomes ay ang ating mga DNA. Tingnan natin ang mga structures ng DNA kung ipagsasama-sama natin. So ang DNA na nasa loob ng nuclei is wrapped or binabalot na tinatawag na histone protein. If we pack it tightly together as it replicates, we form chromosomes. Bawat sister chromatids ay exactly the same.
So we have 1, the DNA, 2, the histones, 3, 4, and the 5th is the sister chromatids.
Tingnan ninyo ang structure na nakadikit sa gitna ng ating mga chromatids. Yan ang tinatawag na kinetochore.
So this is our chromosome with sister chromatids, and in the middle are the kinetochore. The green are basically microtubules.
Ang mga kinetochore ay mga protein on the center of each sister chromatids which will attach to microtubules. We will see that later.
So basically, this is our chromosome, group of chromosomes. Nothing is happening at this point, and then it suddenly condenses, and then the nuclear envelope disintegrates there, and there were spindles separating the chromosomes into 2.
So, as you can see, we have more chromosomes duplicated. The chromosomes started to condense. Microtubules now are attaching to kinetochore, and some of them go all the way across the cell, hindi tumama sa kinetochore. Next, nakikita natin that chromosomes are now all lined up. Nakalinya na sila. Sister chromatids are pulling apart, naghihiwalay na sila. And now, on this part, they are going to each of their individual nuclei. From condensed chromosomes, they loosen up again. Ngayon, balikan natin ang lahat ng steps. Ang unang phase ng cell division ay ang interphase. So, sa interphase, walang masyadong nangyayari sa interphase. So, kung babalikan natin, sa early phase ng interphase, kinopya ang DNA or DNA was duplicated. At sa late phase ng interphase, ang mga centrosomes were replicated. From one centrosome, we now have 2. So, tingnan pa rin natin ang cell as we move on to the next phase, which is the main phase, mitosis. Sa unang stage ng mitosis, which is the prophase, may dalawang malalaking pagbabago. Ang first ay chromosomes condense. Nagkakaroon ng pagsisikip, pagdami ng mga chromosomes. Second, the mitotic spindles form. Nagkakaroon ng mga mahahabang spindles or stick-like structures. Katulad ng blue na nakita natin sa video natin, ng videong pinanood natin about sea urchin cell division. Ang sunod na phase ay ang PROMETAPHASE. Sa PROMETAPHASE, the nuclear envelope fragments. Ibig sabihin, nawasak ang nuclear envelope, and microtubules are attaching to the kinetochores in the middle of the sister chromatids, like what you are seeing now on your screen. Not all of the spindle, of course, would be attached to the kinetochores. In the metaphase, we call this phase plate or metaphase plate, where chromatids line up in the middle. The next phase is anaphase. What happens here is that ang chromatids are separating anaphase from the letter A, like apart. I think pwede yung tandaan yan. Anaphase with an A means apart. So the chromatids are separating apart with equal amount of DNA for each daughter cells. Ang huling phase naman ay tinatawag na telophase. So in our discussion today, I included or I put together telophase and cytokinesis. What happens here is the cell elongates. That means humahaba yung cell. And there is a formation of cleavage furrow, the one you see in the middle, and the daughter nuclei also form. Yung mga ibang microtubules na hindi na-attach sa kinetochore natin ay nagsisilbing guide naman during elongation o yung humahaba na ang cell bago ito mahati. At syempre, importante din sa phase na ito ay yung pagkakaroon na natin ng dalawang bagong nuclei. Isa, syempre, sa bawat daughter cell. Saan naman galing ang kani-kanilang nuclear envelope? Natatandaan nyo yung nuclear envelope na nawasak? Sila ay muling bumalik kasama sa paghati at muling ginamit ng mga daughter cells sa kani-kanilang sariling nucleus. Kung titingnan natin, ang susunod na phase ay babalik ulit tayo sa interphase. At syempre, may mga bagong cells tayo. Ibig sabihin, ang ating mga bagong cells ay mag-uumpisa uli sa cell cycle. Ang phase na yan, kung saan sila mag-uumpisa, ay tinatawag na interphase. Okay. So, kung titingnan again, pwede natin gamitin ang acronym na ito. Meron tayong IPPMATC. Ang I para sa interphase. Tandaan lamang na ang interphase ay hindi bahagi ng mitosis. Ganon din ang cytokinesis, yung C sa huli. So, kapag pinakita sa inyo ang itsura ng cell habang ito ay nagdi-divide, madali nyo nang makikilala kung aling phase sila nandoon. Ang I, again, ay interphase. Ang unang P ay PROMETAPHASE. Ang P na pangalawa ay PROPHASE. M for metaphase, A for anaphase, T for telophase, and C for cytokinesis, which we know is not really part of mitosis. Tandaan lamang na ang interphase ay hindi bahagi ng mitosis. Ganon din ang cytokinesis sa huli. So kapag pinakita sa inyo ang itsura ng cell, ito. Habang ito ay nagde-divide, madali nyo nang makikilala kung aling phase sila. Halimbawa, kung titignan ninyo ang pang-apat na cell, the fourth cell. Yan ang pagse-separate ng mga chromatids. Maaalala natin ang salitang apart or separate. Dahil ito ay A, ang phase na yan ay nasa anaphase, if you can remember the trick. Ngayon naman, bibigyan ko kayo ng panahon para itry nasa anong phase ang cell number 3. Ang sagot ay metaphase. Sa phase na ito, ang align or nag-a-align ang mga chromatids sa middle o gitna. Very good. Ngayon ay siguradong handa na kayo para tukuyin ang mga specific phases ng mitosis na nangyayari sa maraming cells, di lamang sa katawan ng tao, ibang hayop at mga halaman, kundi pati na rin sa mga bacteria, protists at fungi. Bago tayo magtapos, I'd like you to get a pen and a paper and try to identify the different phases our cells are in. Please pause the video if you need more time. Alright, I hope you were able to answer all the items correctly. Thank you for your time and I hope to be with you again soon. Please hit the like button somewhere here below this video before you go. Goodbye everyone.